Cell division is a basic division of the cell. We're going to call this the cell cycle. We need to understand and remember what we learned in the cell chapter. We have to remember eukaryotes. Eukaryotes are different from prokaryotes. A eukaryote is an animal, plant, fungi, or protist because these living organisms have organelles. So eukaryotes have organelles such as the nucleus. Prokaryotes, no organelles. Now both of those do have DNA, but eukaryotes have organelles, prokaryotes, no organelles. You should remember and know what the nucleus is as it contains the DNA, as we've been talking about what the nucleic acids are, the DNA and RNA. To look at the DNA a little further, we're going to break apart and look at the structures of the chromosome. A chromosome looks like a little X, but really what it is, it's just the DNA winding up into this pattern, into this little X shape. Now normally the DNA is all kind of tangled or all loosened up in the nucleus, but when it's getting ready to go through cell division, it's going to wind up. And this is something that God has designed to keep everything together. So the chromosome basically is just the strand of DNA that's usually found in the nucleus of the cell. Now if you look at the picture there are two parts. There's a left side and a right side of the X. The left side and right side are called chromatids. So having two of them, they are called sister chromatids. Now what holds the little chromosome together, that little pinching area, that is actually called the centromere, and they're going to be pulled apart by something called spindle fibers, and we'll see that later on. But the general name for DNA, you can see on the right side, is called chromatin. So if I ever use or you see the word chromatin, just like the word chromosome, it has chro in it, okay, C-H-R-O. Chromatin just means DNA, and a chromosome is wound up DNA. So let's take a look at two terms you need to know. The first one is a gene. A gene is a segment of the DNA. So it's just a section of the DNA. That's all it is. So if you ever heard of someone says, okay, I'm like my father, it's in my genes. Basically what they're saying is my genetic character traits, I'm like my father. Okay, so the segment of the DNA. It's not the whole DNA, it's just a section. Then we're going to be talking about heredity later on. Heredity means passing on these genes or these traits. So if you have black hair, you might have got black hair from mom or dad. That is the passing on of the traits. That is called heredity. Okay, we talked about a centriole very briefly when we talked about the organelles. This is where the centriole is very important. We talked about the definition of centrioles very briefly, and that was describing cell division as basically these little microtubules, these little tubes actually come out to aid in this process of cell division. You can see they're on the poles, okay, kind of like a North Pole and South Pole or East and West. So they're going to be on the poles, and what they're going to do is they're going to pull these little strings, these little ropes, and that will rip apart the chromosomes. Now centrioles we said are found in the animal cells, and these are going to be more of the shorter microtubules. Now this is something very important. A karyotype. Look at the bottom of the slide. A karyotype is a chart that shows all the chromosomes. Okay, if you look at each of the sets, actually, basically when you look, you have the number 1. There are 2 chromosomes there. It looks like that there's actually just one X, but it's not. It's actually 2 separate chromosomes. And so if we go through and we count, basically there's a total of 23 chromosomes. So you can see 1 through 22, and then you have the last one, the 23rd. Okay, the 23rd is either the X or Y. So basically that is going to be the sex chromosome, which means it identifies or controls gender. So if you have an X and Y, that's going to be a male. If it's an X and X, that's a female. So there is a total of 23 pairs of chromosomes. So that means there's a total of 46 chromosomes. So 23 pairs, a pair means 2. So this is 23 times 2. So there's a total of 46 chromosomes. So if you look at this again, you can see there's a total of 22 pairs of regular chromosomes plus 1 pair of the sex or gender chromosomes. So a total of 23 pairs, which is 46 chromosomes. Actually, you're going to get 23 from mom and 23 from dad. And then when you are born, you have 46. So we'll be talking about that later on further in genetics. So let's look at the cell cycle. There are 3 main steps. There's the first step, which is called interphase. In interphase, there's going to be growth and copying of information. This is going to be the longest phase. Then we move into mitosis, which is the actual splitting. Okay, the actual splitting of the DNA and the ripping apart of the chromosomes. And then the last one is cytokinesis. Now, something you need to know, cyte, C-Y-T, means cell. Cytokinesis means cell pinching. So this is when the actual cell is dividing. The cell cycle contains 3 steps: interphase, which is cell growth, copying of DNA, and more growth. Then we have mitosis, which is the ripping apart of the chromosomes. And then we have the actual cell pulling apart and separating, going from 1 cell to 2 cells. Okay, so let's look at the cell cycle. The cell cycle focuses on growth, copying, and then a little more growth, and then division. The cell cycle consists of those 3 phases. Basically, the first step or the first phase is interphase, which is growth, copying, and more growth. Then basically it moves into mitosis and then cytokinesis. So let's take a look at, here's the whole cell cycle. You can see that interphase is majority of the cell cycle. We have a lot of growth here. Then we have copying, which is synthesis. Then we have more growth. And then the very small part is actually mitosis and cytokinesis. So interphase, which is the first step, the focus is going to be growing. Because that cell has to divide, we don't want to keep it a normal size, because if we keep it at a normal size and then it divides, it'll be smaller, right? So the cell has to get bigger and grow and stretch. It gets bigger so that when it divides, it will be the same size as the original cells. So we're going to divide these or rename these growth and copying stages by some abbreviations. So the first one, the first growth phase, think of G for growth, G for gap. We're going to call it the G1 phase. Think of the growth 1 phase or the gap 1 phase. So that's going to be the first one. The second step of interphase is synthesis, the S phase. So S for synthesis. Synthesis means copying or replication. So we're going to take that and we're going to copy. We're going to double it. We're going to double the DNA. So now it looks like little chromosomes, little X's. Okay. Then we move into another growth phase to get it a little bigger. Okay, so the formation of the chromosomes are actually at the synthesis stage. Then we move into mitosis. Now we're going to talk about meiosis later on, but let's focus on mitosis. Both of these are a form of cell division, but mitosis is going to be the focus on the somatic cells. When I say somatic, look at the word to the left of the word somatic, body. These are the body cells, such as bone cells, muscle cells, skin cells, brain cells. Okay, so mitosis is the division of the body or the somatic cells. Well, meiosis is going to be also a division, but this is going to be a division of the sex cells, such as the egg or ovule or the sperm or pollen. Those are going to be the sex cells. So those will actually have to go through a double division, and we'll learn about that later on. But let's first talk about mitosis, the basic division of the cell. One way to remember this, because in mitosis there are 4 steps. So we have, right before mitosis, we have interphase. We see that, but that's not part of mitosis. But now, once we start with prophase, that's the first step of mitosis. So the 4 steps are prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase, or PMAT. You can remember this by put me all together. Or you can come up with your own mnemonic device. Okay, here we have interphase. The cell is getting bigger, it's growing, it's copying, it's duplicating. We have the synthesis. So this is where we have G1, S, and G2 in interphase. Now let's go to the first step of mitosis, and this is prophase. So at the beginning of prophase, the little chromosomes start to form, and the centrioles, those little microtubules, move to the poles. At the end of prophase, okay, the chromosomes form their shape, they shorten, and then basically we have these little ropes coming out of the centrioles called spindle fibers. And you can see the centrioles are at the poles. Then we move to metaphase. One way you can remember this is M for metaphase, M for middle. The chromosomes line up in the middle. Okay, the chromosomes line up in the middle, and the center of the chromosome links up with the spindle fibers, those little ropes, because those little ropes, as you're going to see in this next one, pull and rip apart into those sister chromatids. In anaphase, they get ripped apart. The chromosomes get ripped apart. Then we have telophase. Telophase is the last phase of mitosis. This is when there starts to be a pinching, which is the start of cytokinesis. And then you're going to start dividing, and then you end up with 2 cells. So you start with 1 and you end with 2. So that is cell division. You can see right here, this is an example of metaphase. You can see the chromosomes line up in the middle. The centrioles pull apart, and here's a microscopic picture of the centrioles and the spindle fibers, and you can see the chromosomes in the middle. Okay, here's a 3D model, computer-generated model. And here is cytokinesis. Cytokinesis is describing the pinching and separating of the cell. There's a word for an even splitting or even pinching, and that is called cleavage. When you go to 8th grade and you learn about rocks, if a rock breaks evenly, that is called cleavage. So an even splitting or separation of something, in this case the cell, that would be called cleavage.